my big goal in life has always been to come up with like a unified model of protein evolution that accounts for all these different effects. And so what may appear to be creativity is just trying to tackle every part of the problem. But I think one thing that maybe computer scientists don't quite realize yet is that all of biology is related. Like every biological data point, there's no IID. There's every sample is related to another sample out there. And so if you do like a random train test split, you might actually have overlaps. And in some ways that you could think of that's what essentially Alphafold's doing. Like Alphafold say, I'm going to make a guess that's like 0 recycles, and then you iterate and you sort of move around. But maybe if you do many independent seeds, and I think that's what some of these models like O1 and O3 are doing. Like they have many independent starting points and they explore. And so I think in some ways, I guess we could say we've been already doing that for a while in the protein world. And they're kind of catching up. I've never sort of sat down and said, okay, this direction is probably the most meaningful thing to do. It's more just like, okay, this is like a puzzle and there's no solution here. I'm just trying to figure out what's going on here.
Today, I'm going to be talking to Dr. Sergey Ovchinnikov, a recent biology professor at MIT. Sergey is an easily recognizable name to those in my own field, as he is one of the undisputed greats in the world of machine learning-assisted protein engineering. His prior research includes ways to make protein folding more accessible, models that can generate de novo protein binders with massive success rates, and methods to help scientists learn what protein language models are actually learning about protein folding. Today, we'll be talking about issues with existing protein models, what future protein models may look like, Sergey's own journey in this field, and lots more. Thank you for coming onto the show, Sergey.
Yeah, thank you so much for inviting me to be here. Excited to talk to you about those topics.
Yeah. And so just to start us off, briefly, I'd love for you to give us an overview of what your historical research focus has been and what types of problems you're most curious about.
Well, I would say my journey started in trying to understand the relationship between species, how different organisms are related to each other, but more specifically trying to compare their DNA sequences, their protein sequences to each other. And one of the things that we encountered early on is that sometimes you might think two things are highly related to each other based on the similarity of their protein sequences, but in fact, it's because of convergent evolution. Like, so there might be similar selection pressures in two different organisms that make their protein sequences look very rich similar, when in fact, it's essentially just convergent, like there's similar selection going on. And so to be able to separate that, you really need to understand the underlying protein structure and also understand the protein function for these protein sequences. And so my journey started off actually in phylogenetics, and then I transitioned during my PhD to try to say, okay, I need to learn everything about protein structure in order to sort of go back and maybe correct the signal so we could do more proper phylogenetic analysis. And in some ways, I'm still trying to do that. Like that's one of the areas we're actually pursuing. Like we're thinking about how do we do better phylogenetics. But along the way, we've did a few side projects, you could say, like try to, hey, maybe get into protein design, but that's all sort of related to building models and evaluating these models for this, I guess you say, general goal of understanding protein evolution and evolution in general.
Do you, it's interesting that your original background's in phylogenetics, and you're still like really curious about phylogenetics, even though you're probably most known as being the protein design guy.
Yeah.
Do you plan to do that many pure phylogenetics projects in the future, or is that kind of like on the back burner for now?
You know, we do have a few projects actually going on in the group where we're trying to actually do better phylogenetic reconstruction. But even for the reverse problem, so one thing I've always thought of it as sort of like, okay, structure's getting in the way of phylogeny, but now we're also beginning to believe, like, for example, closely related species could actually influence extracting covariance signals, coevolution signals from multiple sequence alignments. And so it's also becoming an issue where maybe for bacteria, it turns out it maybe is not a big issue because most bacteria are highly diverge and you can almost think of it as a star tree. They're all equally separate from each other. But when you start to deal with eukaryotic organisms, like even fungi, you may have random mutations that are propagated and might mislead some of the calculations in terms of structure. So in some ways, not just purely trying to understand phylogeny, but turns out phylogeny might actually become an important thing to think about when doing even studies more on the structural side or even protein design side.
And for people who like aren't super aware of like how Sergey's work translates to like the current state of the art in the field, there's a pretty clear direct line between the coevolutionary work you did in your PhD and a relationship between that and how a model like Alphafold 2 actually works. In many ways, you were dramatically ahead of the curve. I'm curious, during the mid-2010s, when you wrote those papers, was it clear to even you that this work would be particularly useful for the problem of protein folding? Or did it feel very much like a pure phylogenetics problem that had like no relationship to any like translatable research?
Well, I think in the early 2010s or so, um, there, I mean, other folks besides myself have been thinking about, could we somehow extract covariance signal, and could that be used to predict structure? So I think that's always been on the radar in terms of people like this convolution signal will be useful for structure prediction. Um, at the time, I was mostly thinking from the perspective of using that signal to subtract it out and do better phylogeny. So that was my initial goal. But one thing we've found out is that actually the signal is actually useful, especially if you start to look at metagenomic sequences. And what I mean by that, one of the interesting things people have found was that when you start to ask the question, okay, where is coevolution useful in terms of being able to predict contacts, to be able to predict structure, often those structures are already solved. Like somebody's already determined that structure because if there's a lot of proteins for that protein family, very likely that somebody's already, one of those sequences, somebody already predict the structure for, actually determine the structure experimentally. And the only things that were left unsolved were membrane proteins, because those were really, really hard to crystallize. But with metagenomics, what happened was suddenly protein families that had only few sequences suddenly had a huge amounts of sequences. And so now these coevolution methods suddenly became more and more relevant for those kind of protein families. Yeah.
I remember when we first talked a few weeks ago, you said like coevolution for the problem of protein structure prediction was one of a few other parallel directions that were going on. I think you named two others. I'd love to hear you recapitulate that, because I think that was a really interesting story.
I think maybe I brought up the fact that, well, maybe I talked about like different groups working on this. Is that what you're referring to?
Maybe.
Yeah. Maybe that's what I was. Okay. Yeah. So essentially, I mean, the idea of extracting coevolution from multiple sequence alignments has been around for a while, and actually turns out this still continued to be pursued in three different fields. Essentially, like people in the physics world work with things like POTS models and saying, or icing models, like how we transfer this to this problem. People in computer science world were also thinking like Markov random fields, Boltzmann machines, like how do we use that? And then people in the, I guess you could say, more computational biology field were also thinking along those lines, like mutual information and applying those kind of approaches. And I think the part that I found remarkable or interesting looking back is that often these people didn't cite each other because they didn't know about each other. Like it turns out like if you actually look at the math is almost identical, but they just never sort of talk to each other because they use completely different terminology for the same concepts.
But the underlying like data they used was all identical. Like
the data was used, even the algorithms were the same. It's just that I think in computer science, people call it Markov random fields. And then in physics, people call it like POTS models. And then other places were just calling it like coevolution models. But it turned out a lot of the math was actually identical. Like they just use different symbols. Like one field would use like W to represent coevolution, other would use J, and it's like, okay, but if you look at it, it's like it's the same equation. They're just using different symbols, different words for the same thing. But I think on my side, I worked more, I guess, with the folks coming from the computer science side. So Heitu Kemisetti from CMU in Pittsburgh, and so I came from the Markov random field perspective, and I think in our papers we always call it like Markov random field. But then I realized sometimes people who do POTS models, they get a little confused. Because they're like, hey, is this something different? But it's like, no, it's the same thing. So now I have to say both terms when I refer to it.
Do you think like the field is generally consolidated into like the pure computer science direction, or like there's still computational biologists and physics people who are like pursuing their own like parallel paths?
I think they've all sort of, I think past 2011 or so, when sort of things suddenly started to work, I think all these groups sort of became aware of each other, and some folks actually started collaborating with together. And so there's been, so now I think they're all kind of aware of each other, but I say pre-2011, it's been kind of multiple parallel efforts of maybe people not recognizing that they're working on the same thing.
And speaking on the, like your own computer science background, there's relatively little publication history on your own backstory and like the lore of Sergey. I'd love to hear about why you decided to study biology. What made you focus on phylogeny? And what led to the eventual pivot closer to computer science applied to the whole subject?
Uh-huh, uh-huh. Um, let's see, how far should I go back here?
We maybe even start in college or maybe even high school if you think that's.
It's a good question. So I started off back actually when I was still in high school. I was on robotics teams. I used to program robots to be able to, there's this thing called US First. And it's essentially a team of, I think it's an international now, but at the time they were high school students would build robots and they would compete with each other. And I was the one usually involved in programming these robots. But the reason why this sort of led to biology is, to me, when I started to think about biology from the perspective of code, it all clicked for me. And so what I mean by that is you can think of all organisms, they all have some kind of code, or you can think of all organisms, maybe robots to some extent. There's some code that essentially codes for how they act, how they're developed, and so on. But to me, it kind of felt like we didn't really have a good understanding of the compiler that compiled that code. Like, we have all this code, we don't know the syntax. We knew roughly where proteins start and end, but no idea at the time. And so to me, so that's why I got really excited about biology, because I was like, hey, this is unknown code. We have no idea what it's doing. What if we start to compare these codes to each other? And so right now, when we think about GitHub, we could probably look at some project and ask, hey, everybody who's cloned this project and who's modified it, you could check to see which parts were modified and parts were not modified. And that would quickly tell you, okay, the parts that were probably not modified are important for this overall project, and parts that were heavily modified, those parts are probably not as important. Or maybe they were modified and they've gained new function, I guess you could say. And so in some ways, when you're comparing different source codes to each other, to me, it felt like, oh, you can compare a bunch of genomes to each other and figure
this
out what
naturally leads to phylogeny.
Exactly.
Yeah.
So you can start to compare all these genomes to each other. I guess you could say reconstruct the GitHub history of all, I guess you say, genomes. But then that lets you start to understand the syntax and so on. And so that's sort of what got me initially. So then when I went to college, I was like, you know what, I'm going to learn about biology. At one point, I moved a little bit into history, because I was like, maybe I'll do history of science, because I wasn't sure if I was good enough to do science yet at that time. But then eventually I transferred and started doing more biology. But over time, I joined a couple of labs, like one lab that worked on milkweeds, another lab that worked on various arachnids. I was actually participating in extracting the DNA, sequencing those guys, and then getting all the sequences. But once we started getting sequences back, I think my advisor at the time quickly realized that I had some computational skills. So okay, maybe you could help assemble some of these sequences. So I worked on genome assembly and building some algorithms to be able to do that at the time. And that sort of, I guess you say, brought me back into computation. Because initially I was like, I'm going to do biology, and then I'm now back to using these algorithms that I worked on in the past.
Mm-hmm. And from there, you also did your PhD work in phylogeny.
Oh yeah. Maybe I should clarify. So for my undergrad education, I joined a couple of different labs that were working on phylogenetics. Mm-hmm. But then for my PhD, that's where during my undergrad, I started to realize we can't really do correct phylogeny without understanding structure, without understanding covariance patterns. Because one of the things that's interesting in phylogenetics is when you compare a bunch of sequences to each other, there are certain sites that have high entropy and mislead phylogeny. And what I mean by high entropy, there's certain positions that just change rapidly. And there's certain organisms that also evolve rapidly. And those organisms that evolve rapidly, they would appear just by chance to be highly related to each other. Like there's this process called long branch attraction in phylogeny, where highly evolving species suddenly start to get grouped together. And so to get around this problem, what folks sometimes do is they would say, okay, let's remove positions that are not consistent with each other. Because if there's multiple positions that are kind of consistent, then you would say, okay, this is probably due to phylogeny. Positions that are inconsistent, those are probably just due random. And so we should just remove those. But it turns out these metrics of looking for these self-consistencies between positions is actually very, very similar to coevolution. Like these are sites that are covarying with each other, but it's not clear, is this covariance due to phylogeny or is this covariance due to coevolution? And the only way to tell that is to say, can we look at the protein structure? Like if those two positions are consistent and they're close together in the structure, that's a strong indicator that maybe there's some coevolution going on, and that could mislead your phylogeny signal.
Yeah.
And so it sort of turns out these signals are completely entangled. And so that's when I was like, okay, I'm going to go to get my PhD, I'm going to learn everything about structure so I could disentangle this effect. And that was sort of, I guess you say, my journey into that space.
And then you graduate for your PhD, and during your postdoc, you continue this exact same line of work.
Yep. I guess now that I felt like as an undergrad, I understood phylogeny. Then as a grad student, I understood structure. And I was like, okay, now I'm going to combine the two and finally try to resolve this problem. And so then I went on to become a fellow at Harvard University. And there I actually started to say, okay, how do I actually combine these things? Like, how do I build a unified model that understands conservation, coevolution, phylogeny? And that was my work during, I guess you could say, during my fellowship. But during that time, large models started coming out. Like, for example, folks started training giant language models for proteins. Things like initial versions of Alphafold started coming out. And there I was thinking, okay, is it possible these models have already learned to do that? Like, did we kind of get scooped without realizing? Like, are these models learning phylogeny? Are these models learning coevolution? And so in some ways, my work kind of partially pivoted towards like, let's fully understand what these models are actually learning. Like, we really need to dig into them, because that would tell us, one, did we get scooped? And two, do we still need to work on this problem? And if we are, I guess, scooped, I guess it's the same one. But yeah.
I'm curious. I think you're often, whenever people like think of Sergey Ovchinnikov, they often think of like deeply creative papers. I think papers that you wouldn't really expect to come from any other people. Do you think there's an aspect to do you think a lot of your quote unquote like alpha as like a researcher comes from your background in phylogenetics, and that like people who work at like isomorphic and evo scale could like stand to learn a little bit more about phylogeny?
I don't know if that's where it's coming from, but I guess for me, I guess my big goal in life has always been to come up with like a unified model of protein evolution that accounts for all these different effects. And so what may appear to be creativity is just trying to tackle every part of the problem. Like, for example, we're trying to extract coevolution signal. But then we also need to think about alignments of sequences, right? So for example, maybe we're extracting the wrong coevolution signal because sequences are misaligned. And so we venture into the alignment problem. But then once you start thinking about alignments, then you're like, well, how do you know you got the right alignment? Well, that's where he's like, well, maybe a structure prediction model could tell you that the alignment's correct or not. Right. And so I guess what you could say, what may look like creative, it's all just trying to solve this unified model problem.
I guess like
how would be one way to put it.
How often do you like return back to your background of phylogeny when you're like looking at these problems? Are you often thinking from like a phylogeneticist lens, or are you often thinking from like a pure machine learning researcher lens?
I guess I'm always thinking from the, I guess you say phylogeny or protein evolution lens, would be one way to put it. It's so I'm always coming back to this, thinking like, how do we solve problems in this bit, like constructing this unified model that I keep talking about.
And like one of the axioms you have in your head when you're working on these problems is that you can learn almost everything you need from conservation. Is that fair? Is that a fair way to put it? I do notice you have this like side interest in like molecular dynamics. I sometimes see you like post papers in that realm on your Twitter, but you don't actually like ever seem to like publish in that area. Do you think molecular dynamics will actually become really important in the future when like Alphafold 4, 5, 6 comes out, or do you think like for the moment conservation is like the most important thing?
Let's see. But I guess when I say conservation, what I mean is like there's certain, I guess you could say there's certain very important positions that are highly conserved for purposes of because of function. So maybe to step back a little bit. So we have, I guess you say there's lots of sequences out there, and every sequence has some amount of selection going on. And some sequences are maybe and one particular organism, and that organism needs to do some particular function. And maybe a group of organisms, they all have the same function, and so they have a highly conserved position. But it's not because there's coevolution there. It's just that position is super, super important for that group of organisms. But it could get misleading from the perspective of phylogeny, meaning like maybe you have some random mutation that happened early on in the tree of life, and now because of this doubling effect. Like you have one speciation event and now you have another speciation event. And every single time, I guess you say, half the organisms now have that random mutation. And that could be a little bit misleading for a perspective of saying, is this really conserved or is this just a signal that gets propagated and propagated? And so sort of decoupling those effects. But maybe coming back to your question of molecular dynamics, I mean, it's one of those things where we do like when I guess when we're interpreting models, we're thinking about like how are these models working? Like for example, like language models. If we step back to a language model for a second, are they learning conservation? Like learning each group of sequences have different levels of conservation just because they belong in certain space of sequences? Are they working because we have coevolution going on and different groups of sequences have different coevolution? Or are they somehow internally in the model, like solve the protein folding problem, or maybe doing molecular dynamics? Like is each layer sort of learning at different steps of folding or physics? And when these first models started coming out, it was a little unclear which of those would be true. Like is each layer sort of folding up the protein? Or is it sort of picking up on all these statistics of evolution, I guess you could say. And so that's one of the problems we're trying to separate. But that being said, of course, if we want the model to learn physics, then maybe we need to get into molecular dynamics in terms of trying to get these models to reason over molecules, reason over interactions of atoms. I'm not sure if that's what you're asking or getting to, but that's
I guess the broader strokes of the question is, do you think we'll like ever escape like the well of having, because like you published this paper last year about how this like pattern of a model always learning coevolutionary statistics even continues if you don't have multiple sequence alignment, even if you have like a pure language model that has only seen sequence and like no conservation, it still is like learning folding via evolutionary statistics. And we obviously care more about like the scope of all possible proteins rather than just the proteins that are like near evolutionary proteins. Is this like a problem in your mind that like we need to find some way to move beyond like the well of evolution? Or do you think for now it's like actually like completely fine?
Oh, I think it depends on what you're trying to do and what your claims are. And what I mean by that is, like, I mean, I guess from the pure engineering perspective, there's probably nothing wrong with say, hey, let me grab a piece of evolution here, piece of evolution here, and sort of stick it together and make some kind of Frankenstein protein. And I think protein language models actually seem to be really good at that. They've learned different parts of proteins, which proteins tend to be the same, which motifs tend to be the same across many different proteins. And you can imagine using such a model to sort of stitch different things together. But then, of course, for somebody who's maybe coming more from the first principles point of view, they're like, hey, this is kind of cheating. Like we want to be able to understand why these sequences code for these things and why are we able to stitch these things in a certain way. And maybe that would allow us to move into space that nature has never explored. But then again, there's people that argue also that maybe any protein seems to be a combination of fragments. And so maybe nature's already explored all possible fragment space, in which case having a model that learns fragment is not a terrible thing. You just need to know, you just need to at least just sample these recombination of things.
Which do you think is true?
I think there's definitely space still left to explore. The way I like to think about it is that there's any given protein sequence is sort of composed of ambiguous and non-ambiguous regions. And so what I mean by that is like there's certain regions that there's like a sequence, it always codes for a helix. There's a sequence that always codes for a particular turn. And there's parts of the sequence that these ambiguous motifs that you have actually no idea what they're coding for. Like it could be a helix, it could be a beta strand, it could be a loop, it could be a break there, so on. And the only way you'd know is in the context of the fold protein. Like the tertiary structure folds up, and you're like, okay, it's almost like this region is some kind of chameleon sequence that can sort of adapt to different things depending on its context. And I think any natural protein is sort of a combination of these two things. There's certain parts that you say, I want this part to be rigid. And so you probably want to have like these non-ambiguous sequence. And then there's some region that are, you say, you know what, maybe this part needs to be flexible. And so maybe I'll put in some ambiguity here that maybe could respond upon a ligand coming close. And what that means is that now, of course, if we try to predict these proteins, things that are made up of purely non-ambiguous sequences, Alphafold, ESM fold, like all these fold methods can predict them really well because I think they've already learned all these sort of non-ambiguous sequences, motifs that all these proteins share. But then for regions that are more ambiguous, that maybe to have to do with function, these models, unless there's evolutionary information associated with them, they're unable to predict it.
Do you think the future looks something like you have like one model to predict the like non-ambiguous parts and then like a more physics-based model to predict the ambiguous parts? Or yeah, like I'd love to get like, I guess like the broader question here is what do you think like AlphaFold 4, 5, 6 looks like? Do you think it goes something in that direction or somewhere else?
Probably. I would say that if you construct a protein that's purely from these, I would argue, non-ambiguous sequences, and I would say all of de novo design, people who do de novo design often completely just exploit these non-ambiguous sequences. Like every single stretch, you could actually even do this experiment. Like if you take any little cut and throw it into AlphaFold, it will predict that without the context of the rest of the protein. And I would say it's a very simple search problem. You're essentially just stitching fragments together, and that's why it's really, really easy to predict these proteins. But of course, now once you start moving into this more ambiguous space, it becomes a really large search problem. And there you sort of do need to maybe start to say, okay, maybe we need to put in physics in here. Maybe one way to put it would be like there's this global search problem where you do huge, like I guess you say fold exploration. And then there's more like a local search. Like once you roughly know what the fold is, you sort of fix all little details. And I think AlphaFold has learned to take sort of a few steps along some kind of energy function that it learned. But it has a really hard time doing a kind of global search.
Like I remember you said like there's this figure in one of your papers where the AlphaFold uses the MSA to find itself like roughly where it is on the energy landscape, and then like does some local energy minimization from there on out.
Exactly. So I guess our current hypothesis is that multiple sequence alignments sort of give you the global or give you like sort of, I guess you could say you can skip the global search and you're just focusing on the local search. But sometimes you can mess with the multiple sequence alignment. Like people have found, like if you subsample the MSA, sometimes you turn on dropouts, you enable sort of random masking. You could sometimes explore other parts of this starting space. And in some cases, like what we try to do is say, well, what if we just give it like a template structure? So this is work with James Rooney, and we're like, well, we could give it some starting point and try to almost like move around this space and see how well does it actually able to know which part's correct, which part's incorrect. But coming back to your question of like, what's the next version of AlphaFold? I do think it's probably going to be some version where there is some global search exploration that gets baked into the model. And so what I mean by that is like, you can imagine in every instance of AlphaFold is sort of a few moves of a game, but it's not like the full game that we're playing. And so, but there's probably many, many starting points. And so having some way to sort of guide or move into this, I guess you say global space is going to become quite important as we move into space where we say, well, how about proteins where we don't have evolutionary information and so on? Or if we want to design these proteins that look completely different.
By many different steps, could I mentally equate that to inference time compute, or are you referring to something else?
I guess two things here, because so I guess want to step back a little bit. There has been researchers that have shown that you can run AlphaFold with thousands and thousands of seeds, and you could sort of think of each seed as sort of being seeding some independent MCMC trajectory and some search. And so you have a bunch of random starting points. And for example, even AlphaFold 3 for antibody antigen, where there's no evolutionary information, they found they have to go up to like 1,000. And even after 1,000, you can continue adding more and more seeds. And so those are kind of things where those are kind of random starting points. And so if you're lucky, one of those things will get to the right answer. But one could imagine doing something, say, well, what if we have a smarter way of seeding, like some smart seeds? Is there some way to bypass? So I can almost imagine some kind of a model that sort of says, okay, here's where you need to explore, or here's some hypotheses where to explore. And these are some of the directions my group is currently exploring, saying like, could we somehow seed or do some smarter seeding of the space? Yeah.
I think like going back to like what your lab is working on, I think a theme I've seen in a lot of your labs working, people who have collaborated with you, is like a belief that the existing base models are actually really powerful, and if you use them in interesting ways, you get a lot of value out of it. Do you think there's that much value in like pre-training from scratch, or do you think a lot more work could be done with the existing models? And there's a lot of really interesting work that could be done there. Like you have like Bindcraft, which is like, I think for people who haven't even heard of Bindcraft, it's just the ability, it basically allows for you to create de novo protein binders at ridiculous success rates, far higher than anything else that had come out before.
Well, I guess coming back to a little bit the earlier point that I was trying to make is like, so we know that AlphaFolds are highly limited to a certain space. Like there's like, I guess you could say this idealized non-ambiguous space. And so the question is like, well, one question is like, do we even need to move into this non-ambiguous space? Or alternatively say, well, actually, you know what? If we're happy with this space and there's a lot of things to accomplish here, why not just like limit ourselves there? Like why even explore to other things? And so and if we take that philosophy, then I would say, well, actually, current tools are fine. We just like we could just invert Alphafold. That's what we do in Bindcraft. You can essentially use ESM fold to sort of score these sequences. Like we think like all these models has learned this sort of non-ambiguous, idealized, I guess you say, low contact order space. And let's just fully exploit that and just design within that space. And I mean, there's nothing wrong with that. It's just a little bit less satisfying from the perspective of say, hey, what if we want to move into something more complicated? And I guess some people could argue that, and we argue this, is that, well, if you start to move into more complicated function, so what I mean is like, if you're binding to something, we're not too much worried about, hey, does the binder change upon binding? Is there some flexibility to the binder? Like the fact that it's like a rock or like doesn't unfold, that this is fine. Like, so it's like binding seems to be a relatively good application of these like highly idealized structures. But then if we start to move into say like now if we want to design enzymes, now maybe we have to start to understand these sort of ambiguity. But maybe there's some sort of middle ground where you say, I'm going to try to make it as rigid as possible, but maybe destabilize a few little things. And so then it becomes like a local search problem. Maybe that's one thing Alphafold can do. And there's been sort of these combinations of methods that have been coming out, like from David Baker's group, is like, well, we could restrict some spots based on what we believe this is what nature's optimized for. We're going to keep that fixed, and then maybe redesign everything else. And maybe we don't fully understand the part that we kept fixed, but we'll just say that we know it moves somehow. We just have to keep it all in that same spot, but everything else we can redesign and make it rigid, and then that creates like a new enzymes.
If it turns out, at least for like the rigid binder world, if it turns out non-ambiguous sequences are just like kind of intractable for like humanity as a whole to deal with, do you think that's like a big loss for anyone? Is it like a big deal, or is it kind of like it's fine?
Well, I guess it depends why you're getting into this problem. I think there's some people who go into protein design, they're coming more from like I guess you could say more of a biophysics background. They're like, hey, I want to understand this problem. And I guess like often people like to quote Feynman saying like, hey, what I cannot create, I don't understand. And so the idea there is like, you want to be able to, if you could say, hey, my model can make something and it works in lab, then I fully understand this problem. And so there you kind of want to have the model work for the reason that you understand, like the mechanism. I mean, I guess now we're in this weird space where now we could actually make things and we still don't understand it. So it's like, what's going on here? So it's like, how do we sort of check understanding anymore? And I think for those folks that want to understand things on more, I guess you say on a biophysical side, they do want to sort of make it work for the right reason, I guess you'd say.
But for the people who are most interested in like, I want to create a protein for a specific functionality. I don't care if it works. I don't care if I understand how it works or not. Do you think non-ambiguous space is like perfectly fine?
I think for the most part it is, depending on the application. So I would say like for binding, I think we're definitely, don't necessarily need to worry about maybe this ambiguous space.
And for enzymes, it's a little bit more complicated.
For enzymes, that's, I think when, like, for example, I think some of the dreams people have is to like design molecular motors, for example. And then there it's like, well, this is where maybe we can't get away with this hack, or folks, they say, well, maybe upon binding, you have a conformation change. I mean, there has been work where like folks have done like these locker kind of proteins where they use like, you have like one helix flipping out and the ambiguity is mostly in that loop. So you essentially have one helix and then you have a loop, and that loop could potentially have a lot of ambiguity in there and it could get displaced by another helix coming in. And so I guess there's like these hybrid things where you sort of combine things that are ambiguous and unambiguous. And so maybe there's still a lot to push in these hybrid-like sequences. Yeah.
If you look at the problem of at least rigid binder design, do you mentally consider that problem like solved? Or are there still like edge cases where you think there's like a lot of work to be pushed on?
I would say that it, it it doesn't work for everything, and part of it has to do with the fact that one thing we find is like if you take a target that maybe has a little bit like a hydrophobic patch somewhere, um, interestingly, if you just run Alphafold with any random sequence, Alphafold always puts any random sequence near that patch.
Mm.
Um, and and then of course once it goes there, it's not really confident. And as you optimize that sequence, it becomes more and more confident. But then there's some targets like you give AlphaFold random sequences, and every iteration it just places in a different location.
Mm.
Like AlphaFold doesn't see a clear signal anywhere where something can go. And I think those are regions that are more hydrophilic. So for example, if the surface is completely hydrophilic, essentially there unless you already have like a perfect sequence on the other side, by perfect sequence, what I mean is like you have maybe the correct hydrogen bond patterns that can maybe detect one part on the surface. It's really, really hard to optimize.
Mhm.
It's like you almost like need the right answer before you even try to design or optimize. This is where we think maybe methods like diffusion or flow could be useful because you target, say, hey, let's just explore here. While hallucination, you're constantly, it's almost like you're hoping that it already knows where to go before you even start.
Mhm.
And that becomes a bit of a limitation because you want to maybe restrict yourself to a certain spot.
That that makes sense. And actually,
Mhm.
like kind of on that point, I think the last time we talked, you had a lot of really interesting things to say about the value of diffusion versus
Mhm.
hallucination or masking. I'd love to get like your pitch as to why diffusion is like the way forwards.
Well, I guess I wouldn't say diffusion is necessarily the way forward, but it is definitely a step in the direction. So maybe to step back a little bit. I mean, ultimately, the protein design problem is find a sequence that folds into one structure and no other structure.
Mhm.
But also not just folds into that one structure, no other structure, but also where the conformational landscape, or I guess you could say the folding landscape, has as a sort of, I guess you could say, a smooth landscape where you can actually get there. So, because you could imagine you can have a sequence that has very low free energy, but there's a huge barrier they have to go over to actually be able to actually fold into that structure. And reason why we like things like AlphaFold or inverting AlphaFold is because we say, well, actually during design, where at every single step of design, we're testing for that condition. Say, does it fold into that structure and no other structure?
Mhm.
Um, the problem with the methods like diffusion is that you're sort of coming at the structure, the sequence is not yet implemented. But even if you do joint sequence and structure optimization, you still at the end of the day, you're only evaluating that sequence for that one structure that you're diffusing. And you're never asking the question, does that sequence fold into something else?
Mhm.
I mean, of course, you can run it thousands and thousands of times and then after fact, like run AlphaFold on all those sequences and check for that condition. But if the goal is if the end of the day you're going to be using AlphaFold, like why not just use it as the oracle itself?
Mhm.
Like so I guess to just to describe to people who are not familiar, like you can either generate a bunch of sequences and then check for this sort of, I guess you could say, inverse folding property of making sure the sequence only folds into one structure and no other. Or you could say, well, let me just evaluate that condition at every step of design. And in the past, people sort of abandoned this idea of inverting structure prediction model just because it was really, really hard to optimize. Like if you try to backprop through AlphaFold, it gets very, very unstable once you start to only work with a single sequence. And it takes too many steps. And so I guess you say diffusion was sort of invented to try to, or at least not necessarily invented, but was introduced into this field in order to get around that problem of sort of the instability of backpropagating through structure prediction models. Um, but I think now we're sort of getting to a point where we say, actually, you know what? With some of these tricks, like relaxing the sequence representation, um, maybe removing some of the recycles from the model, we could actually do like it's not as expensive as it used to be, and now we could actually maybe move back into this kind of approach.
Like the
Mm.
inversion approach.
Yep.
Okay, so actually you don't like I may have like misinterpreted your original viewpoint. You don't think diffusion is actually like particularly like I guess like do you have a strong stance one way or the other as to whether inversion versus diffusion will like ultimately win as the primary protein design method?
Well, I guess what I'm trying to get at is that I think ultimately it's testing for the same condition. Even when you run diffusion, at the end of the day, you still use AlphaFold as your oracle
That's true.
to
Yeah.
check. And so one could argue like, well, if that's your end goal is to pass the AlphaFold test, why not just use AlphaFold throughout the whole process? Um, and in some cases, and so usually people didn't want to do that just because AlphaFold was really, really difficult to work with, like in terms of inverting it and propagating through the signal. But I think the reason why hallucination has been coming back recently is because people have finally figured out, hey, actually there is a way to use it as the oracle and use it during optimization. But that being said, there's still some problems like what I described a little earlier, where let's say you want to design a binder to a particular location, and you give AlphaFold some starting sequence, and that sequence just doesn't go to that spot. And you could add some hotspots. You can say, hey, I really want to go there. But unless just by chance AlphaFold predicts that protein to be close in that region, there's no gradients to push that sequence in that spot.
Yeah.
And so there's just no way to tell AlphaFold, here's where I want to explore. Just you're almost hoping just by chance it appears there, and then you optimize there. In the case with diffusion, since you're working explicitly in structure space, you could say, hey, I have my structure. I'm going to initialize my noise here. And now you're kind of forced to explore there.
You can guide the diffusion process like the tiniest bit.
Yep. So I guess you could say you could directly steer the structural components. And that I think could be powerful in that context. Yeah.
Do you think, I saw that you had this bolts inverse design thing that came out recently. Do you think there's going to be like that big of a step up from like Bindcraft is in some sense like using AlphaFold 2 as an oracle, this like new approach is like using bolts as an oracle. Do you think there's like that big of a step up in actual improvement in terms of generating binders? Because I remember I watched a lecture by Martin a few months ago, and he said like initial tests with bolts actually showed that it wasn't like that much more successful than AlphaFold 2. Does that also continue to be empirically the case?
It's a great question. We haven't really did any kind of benchmarking in terms of protein-protein interactions. Is it able to predict things better? The reason why Yiling in my group has been working on this is we're thinking like, how about other problems where we don't actually have, let's say, proteins that we're binding to. So let's say the protein might be, instead of protein, you say I might have DNA or RNA or a small molecule, or maybe a protein with a modification. And so I guess I was thinking what we call bolts design was more not necessarily because we think it could do better than AlphaFold 2, but more because it could now address other class of problems that AlphaFold 2 cannot do. So anything to do with like small molecules or anything to do with non-protein. But just coming back, just the protein-protein interaction problem, I guess my immediate guess would be that it wouldn't do much better. But that, I don't know. I guess that's something worth exploring.
Yeah. It was surprising to me because I think instinctively I would expect like, oh, AlphaFold 3 has seen more molecular interactions. Thus, it probably learns to do protein-protein interactions better. And it seems like that hypothesis hasn't really proven itself out. Is there good intuition as to why?
Well, I mean, at the end of the day, like if you sort of look at AlphaFold 2 versus AlphaFold 3, and then all the different implementations of AlphaFold 3, like the core parts of the model are the same. Like you still have the Evo former, and then you still in the pair former, which removes parts of the MSA. And the only thing that really got replaced was the structure module got replaced with the diffusion module. But to me, it seems like both of those things are kind of doing the same thing. Like it's just one is a bit more deterministic than the other. Like one, you start with the noise, and then you try to satisfy some constraints coming from the pair features. One, you start all 0, 0, 0 and try to match those constraints. And I guess if I was to guess, I would say maybe the DeepMind team probably thought that by doing the first part and like the more expensive part, we're going to kind of like to say, as sort of a separate component, and then do a lot of diffusion afterwards, like many iterations, that you would get many different solutions. But in reality, what happens often is like the first part just gives you back the same constraints. And so then you always sample the same structure. And so I think there was great plans there, ideas there that might have improved things. But ultimately, I think the structure prediction part maybe didn't get helped too much by this updates. But that being said, like all small molecule, non-protein things definitely improved. I mean, I might take a little part of that back because I think in the Alphafold 3 paper, they did show for like, for example, antibodies, you can do far more far more different random seeds and you could find the right solution. So maybe there is some improvements there. So there, yeah.
Yeah, like on the topic of Alphafold 3, I remember you talking one point, and I might like completely misphrase this, is that you had some quibbles about how the recycling process is being done in Alphafold 3. I'd love to hear you just expand on that.
All right. So yeah. So I guess for those of you who are not familiar, so Alphafold 2, you make a prediction and then you take that structure and you feed it back into the model and you say, make another prediction. You sort of keep cycling this over and over. And each step, the structure kind of improves and improves. And one thing we've noticed with Alphafold 2 is if you try to do like homoligomeric predictions, if you look at recycle 0, all the copies are literally overlapping on each other, completely overlaid on each other. And then each recycle kind of pulls the copies apart. So they're no longer clashing with each other. So what we think is happening is the pair former, or I guess you say the evil former in the context of Alphafold 2, sort of learned some constraints, but these constraints are physically not possible. Like you try to actually realize that structure and you realize that you create a bunch of clashes. And I think one of the cool things about Alphafold 2 is like you see that there's this errors in your structure and you feed those errors back into the model and you refine those constraints and you keep refining it. One thing people have noticed with Alphafold 3 is that you start to see these clashing problems. Like if you try to predict like a homologous structure, these copies are all overlaid on each other. So there's no feedback in Alphafold 3 back from making an attempt to predict the structure and then refining the constraints.
I thought Alphafold 3 does have a recycling component.
So it has a recycling, but it's only within the MSA and pair former.
Gotcha.
Like there's no, like the structure gets predicted, doesn't get fed back into the model.
So there's like almost like an original sin problem going on a little bit where it like has no chance to like try and modify what it predicts.
I mean, yes, exactly. So it's like you try to realize those constraints, but there's never feedback that you fail to realize these constraints. And I think the reason they did this was well-meaning because I think they're hoping like, let's do this whole cycling or this recycling one time and then I don't run diffusion thousands and thousands of times.
Yeah, I mean, the hope is that diffusion does what you're asking for, right? Like it realizes the constraints early on and then refines it. But why do you think? Because like in my head, I've always mentally thought of recycling as like a pseudo diffusion process. How come the actual diffusion process doesn't seem to do what recycling is doing?
I suspect the reason is because the diffusion part doesn't have a chance to sort of remodify how it's interpreting the coevolution signal. So what I mean by that is we think like MSA module and then the evil former or the pair former is taking the evolutionary data and trying to clean it up or trying to disentangle some of maybe ambiguity in the constraints. But once you've disambiguated all those constraints, you're kind of now stuck to just trying to satisfy those finally disambiguated constraints. But what if the ambiguity like didn't work out correctly? Like you didn't correctly disambiguate these things. And the only way you would know is to try to embed those constraints or in a 3D structure. And I guess if one was to go back to the diffusion part and let it reprocess the MSA in each iteration, then maybe that would work. But then you're back to what you call the pseudo diffusion part of Alphafold 2. Because that's essentially what Alphafold 2 is doing. It's like you're doing this iterating process and you're fixing the constraints, refining, I guess you say the potentials, and then repredicting the structure, and you're iterating on that over and over and over. So yeah, but I would say like one simple fix to all of this, say, hey, let's just feed the constraints back in. My guess is the reason why they didn't do this was because of that thing that I talked earlier about these hopes. But that would be one, I think, an obvious thing to sort of say, hey, let's bring back this iterative refinement step. And that would be one way to maybe fix that problem.
Has any of the Alphafold, I haven't looked too closely at any of the Alphafold 3 replications. Have any of them tried to poke at this problem, or they like haven't really touched it?
They have, from what I've seen, they haven't, no.
Okay.
And I think part of the reason is I think everybody right now is just focusing on, say, let's reproduce the baseline. I'm sure that some of these groups are already thinking about how to improve on top of this, but I think right now the goal is to reproduce the baseline.
Yeah, that makes sense. And kind of like on this note of replications, we seem to be kind of entering, like the field at large seems to be entering something similar to GPT-2 moment in biology, or phrased more differently, a period where the best models are being built by private companies and are for the most part never open source. I'm curious to hear your own thoughts about this as someone who has built probably the most popular open source implementation or open source like plugin to Alphafold 2. Where do you think the trend of open source and protein models and biology models is going?
Yeah, I think it's definitely sad to see like companies now to not wanting to release their code, because I always felt there was a synergy between industry who have a lot of resources like GPUs and building models, but often having no idea what the models are doing or how to interpret them, and then academic groups taking these models and trying to interpret them. And so there's this synergy as somebody builds a model, a bunch of people are trying to interpret it, figure out what's wrong with the model, what is it learning, what is it not learning, and then feeding back into the industry, and the industry could try to fix those models. Um, I'm guessing maybe industry have reached a point they're like, say, hey, we've now it works too good. We can make like billions of dollars on this, and so they have less incentive to release these models for academic research. And yeah, so it's definitely. I mean, I'm personally not too happy that they're not releasing these. Yeah, it's
Do you like in the NLP world, like people thought things would go the exact same way, but like you had entities like Meta release the llama models. Do you think there'll be some entity like that in the biology world where some either very strong academic group or some like very, very caring for-profit institution releases good open source models? Or do you think that's also like unlikely?
Well, I mean, like if you look at what happened with Alphafold 3, there have been indeed groups, like I think like ByteDance, for example, released Pro10X. Um, and I think they've now also made it for both commercial and non-commercial use. Chai released their model. And like HelixFold, I think there's been a few groups, I guess, similar to like the whole DeepSeek kind of thing, where they're like releasing everybody for be able to use these kind of things. And so, yeah, I think there's always going to be some groups out there that are just releasing. I'm not sure how much it'll continue. I think sometimes what people do is industry, I'm guessing, is they would release some early versions of things just because it's really good for PR and so on. But then once they get things working much better, they might not want to release it anymore. But then maybe some other group catches up and releases everything. So it's kind of so I think there's going to be cycles where people they think they have something cool and they're trying to hide it, and then somebody else will catch up and eventually get there.
And kind of on this note, at least amongst like NLP frontier AI labs, there's some level of homogeneity amongst all the approaches they're going. Like for the most part, everyone's doing autoregressive models, but there's also like one group doing like diffusion language models because they think like that's how you get like better language generation. It seems like for the most part, almost everyone in the like frontier biology labs are pursuing the exact same approach. From my perspective, do you see like strong levels of heterogeneity in the underlying details, or would you like agree that they all seem to have a very similar thesis?
I think sometimes it looks like everybody's doing the same thing because everybody's just trying to reproduce the baseline. But once the baseline is reproduced, then people start to explore more. So I mean, it's true that like once something comes out, like somebody say, "Hey, we have a working solution," everybody's trying to catch up to it. But once people caught up to it, then people say, "Hey, now how can we improve it? How can we change it?" But every year, you always have somebody else saying, "Hey, there's another way of doing it." And so there's always these shifts, and I guess in our world, we do see like people are doing some kind of MCMC hallucination, and then people switch to diffusion, and now people are moving into flow, and now hallucinations coming back, and I'm guessing there's going to be a sort of, but there's always like some group that shows something, and then everybody tries to catch up to it. So I don't think there's that issue per se.
It does feel like EvoScale is like the most unique amongst everyone by like eschewing MSA. Do you think like getting rid of MSA is going to be like a good long-term solution to not having to deal, because like the MSA tracker is annoying to deal with in practice. But do you think it's like so essential that like it's very hard to escape from needing it?
I guess our argument is that even the models that do work on single sequences, they are learning something akin to an MSA, not necessarily memorizing MSA per se, but the statistics that you would have for a given protein family, it's learning these things. So, and I guess it's sort of a debate between, I like to think of it as sort of a debate of storage versus lookup. Like you could have a model that just memorized all the statistics, or you have a model that's much smaller and can retrieve some information and then using that information, recompute some of the statistics. And in some ways, you could sort of see parallels to that in like the OpenAI field. I mean, OpenAI, where they're often thinking like, hey, instead of making these models bigger, let's just give it access to like Mathematica, or give it access to I think in their case, it would be Bing instead of Google. But it's like giving it access to search. And MSA is actually sort of, I guess you could say, essentially a version of search. Like you search for a few things, and based on that information, you compute the statistics. And so so I guess the question is like, what makes more sense? Do you make models larger and larger, or do you make models smaller and smaller, but give it access to information? And I personally prefer the latter, saying, well, could we just make smaller models that have access to information? But that being said, I think folks that do scale, like evolutionary scaling kind of stuff, I think Alex Rivas and so on, they do believe that as you scale, eventually, maybe at some point, it has some kind of emergent property. Like right now, to me, it seems like it's mostly learning conservation, coevolution, maybe a bit better coevolution in terms of maybe able to share statistics between protein families that we previously were not sharing. But I guess the hope is at some point it snaps out of that behavior, and so it's like, hey, now to make it even better, you need to learn physics. And so that I think it's worth exploring. I mean, it's really expensive. But if we don't believe in emergent behavior, then it's like the question, like why bother scaling? Why not just give it access to a bunch of sequences and let it search for some of those that information itself?
And like one alternative thought, maybe like instead of providing MSA during train time, you provide it during inference time. Like I've seen a few papers try to do like a RAG approach when it comes to actually providing MSAs. Do you think that approach has like much steam in it, or there's some reason it doesn't really work that well in practice?
Sorry, could you explain the process again?
Yeah, like the idea is like you provide the sequence to the model, and the model can like refer to like a retrieval augmented generation database, which contains like embeddings of like homologous or aligned sequences, and it can just like feed that information in, and that's how it computes things.
Yeah. I mean, that seems like one way to do it. I mean, I guess you could always, I mean, to step back a little bit, there's one of the issues with MSAs is that one is often you don't actually know what sequences to include. Like you get a bunch of sequences, but some of those sequences might not belong. Like maybe they have different conformations, or maybe they're completely from a different protein family. In some cases, it's just really hard to find these sequences because maybe they're less than 20 or 10% identical to the query sequence. And so some of the typical methods people used to might not be able to find these sequences. And then finally, there's the problems of just aligning the sequences. And so I guess the retrieval methods could be useful because they're able to potentially say, well, instead of doing sequence alignments to find those sequences, maybe there's some barcode I can use to find those guys. So
like you're probably trading off on something. Like you're losing. Like you're using a heuristic, really. Like you're unable to do the actual alignment, and you're trading off on some level of accuracy. Like
I mean, I think if you try to actually take every sequence and compare it to every sequence, it's just going to take forever. It's going to, like, even making one multiple sequence alignment on like hundreds of CPUs would take probably multiple days to do a search against all the sequences out there. And there's all these heuristics like, hey, let's try find, like, for example, BLAST, the way it works is sort of you chop up things in little words and you say, do any of the words match? And MMSeq does that to a similar extent. And then like ungapped alignments, then gapped alignments, and so on. But you're probably losing a lot of things. And so and you could always find more sequences by turning off all these pre-filtering. And so in some ways, one could also argue that, well, maybe there's just issues in making multiple sequence alignments. And so having a model that just stores evolutionary statistics is better. Or alternatively, maybe there's other ways to retrieve sequences that don't require explicitly aligning things or looking for matching words.
It feels like, I imagine like what some people at EvoScale like think about is, if you try to do the alignment yourself, you can't include like every possible alignment. You have to make some sort of like cutoff as to how many you include. And like how exactly do you do the alignment? And it feels better to have the model decide what is important versus what isn't important. Is it just like, I guess this is like a question more for your phylogenetics background. Do you think the way people handle alignment with models like Alphafold 2 are like actually pretty sophisticated, and there's not that much further optimization that like a zero priors like ESM approach could really improve upon? Or
hmm
you imagine there's like still ways to go to improve things?
I think there's still ways to improve things. The reason I'm saying that is actually in most recent casts, people have found like the best working methods is to essentially try to create multiple sequence alignment in thousands and thousands of different ways. Like people try lots of different methods, different e-value cutoffs, different coverage cutoffs. Um, and you could always make better predictions by sort of sampling lots of different kinds of MSA generation methods.
Mhm.
Or also what you include and don't include. And in some ways, we've also found this before. Like one of the projects we worked on before was say, well, what if you make MSA generation differentiable? Like let's say you have a collection of sequences, but you don't know how they align to each other. Could we pass them through some kind of differentiable Smith-Waterman module and then try to figure out the best way to align them to maximize the PLDDT and Alphafold? And we did actually find that for proteins that have very few number of sequences, sometimes just realigning one sequence made a huge difference. And so there's still room to improve there. In some cases, it worked for the wrong reason. And what I mean by work for the wrong reason is, turns out like this is work from Sam Petty, who's at Tufts now, but she looked at some of these alignments that we generate, like these multiple sequence alignments, like, hey, we got a better multiple sequence alignment, because is it actually better? And it turns out in some cases, the sequence got misaligned, and that improved Alphafold, because right now what happens, this currently aligned sequence was influencing the covariance statistics. And so if you misalign it, it sort of gets averaged out. And so in some cases, some sequences just do not belong. And by realigning them, we actually removed it. And so it's like, in some cases, like, so I guess you could say the MSA problem is still unsolved.
Yeah, like I think like one question that I like have like tried to figure out myself and have kind of come up empty-handed is there a principled way to improve the MSA? And it seems like the answer is no, really.
When you say
Like instead of having
Uh-huh.
to like that like Kasperzeld AF sample where you just like rerun things like a billion times with different MSA subsets, it's like kind of under discussed in the paper as like what makes one MSA better than another MSA. Is there actually like a principled approach to decide which one is better other than just looking at the confidence metrics? Or is there like some like phylogenetic theory that you could rely upon to like prove that like it's clear why this one MSA is better than another?
Yeah. Well, yeah, I guess it's an unanswered question. Like, why does it get better? I guess one hypothesis is like, well, maybe each MSA is essentially initializing this global search. Like, coming back to like the earlier topic, like each MSA is sort of perturbing it enough to start somewhere else, and now you found the solution. And it's not really the fact that MSA is any better. You just add a little bit of noise, and now you're starting somewhere else.
Yeah.
And so maybe if you just use the same MSA with lots of random seeds, maybe we've gotten the same answer. That being said, other researchers like Hannah Wayman-Steele found that actually if you cluster the multiple sequence alignments, you can look at different clusters, and different clusters seems to have different maybe evolutionary signal, and that could push Alphafold to do one thing versus another thing. And I guess originally I would have thought that wouldn't be the case, because with Alphafold, you have a tension between the sequences. And so you're like, well, Alphafold should be able to figure out which sequence to include and not to include. But in practice, it looks like you can get a better signal by essentially sort of moving the signal around and sort of subsampling the MSA to closer and closer to the query. That being said, I think other researchers have shown that actually sometimes you can just make random MSAs and you can still explore that. It doesn't necessarily mean that one invalidates the other. It just turns out there's lots of ways to get Alphafold to predict alternative structures.
Yeah.
Like you can maybe get it to predict the alternative structure because you're just initializing in a different search space, or because there's some bias in one place or another. And so there's a lot of ways to sort of push Alphafold to do different things. And whether or not they're doing it for the right reason or wrong reason is sort of still sort of a debated question, I guess you could say.
I think kind of related to this point, I have been like a lot of your papers kind of poke at the problem of mechanistic interpretability when it comes to these space models. But as far as I can tell, you haven't like clearly tried to like train an SAE on like ESM or anything like that. Do you think there's a lot of like steam in these approaches being applied to protein models, or there's like some gap that you're unsure about, and that's why you haven't tried exploring it yet?
I mean, we have looked more specifically at different attention layers and see does different attention layers maybe learn different things. I guess the reason we never really, I mean, there has been recent papers, I think a couple papers where they look at these sparse autoencoders applied to the not our papers, but other folks have done this now.
like people from in
Uh,
particular, I think also from MIT. They started a company around like figuring out which layers in ESM correspond to the specific secondary structure.
Yeah. I guess, I mean, it's definitely an interesting sort of thing you could look at to see, hey, is there parts? I mean, the reason why we personally haven't done that was because we're like, well, the attention layer already tells us like there's different layers learning different things, and you sort of see secondary structures popping up and so on. So that I guess for us, the attention layers seem to be already interpretable from that perspective. But that being said, maybe there are certain features that are not necessarily in the attention themselves that may also be. I guess it would be interesting to compare these, I guess you could say, sparse autoencoded features to the corresponding, I guess you could say, attention heads, and see, are they picking up something different or not? I mean, I guess to step back a little bit, folks have in the past, like Martin White, for example, have looked at sort of taking these POTS models and decomposing them and finding that different sort of, I guess you could say, components of the POTS model have sort of connection to different features of the protein. So I guess one thing that might be fun to explore might be to say, well, what if we just take like these categorical Jacobians from a POTS model, I mean, from a language model, and decompose that and see, do these correspond to similar kind of features that like SAA models are picking up on? And I think there's still room to explore that. That being said, I think that the one exciting thing about these sparse autoencoders is that perhaps if you could figure out exactly in the model what is being activated to make a specific prediction, then that could be some kind of steering approach. Like you could say, well, now I know like if this is activated, it means that this is going to be an enzyme or something, or this is going to be a secondary structure. And so then maybe during hallucination, if you want to use that approach, or maybe you could somehow like enable those things. And so if you could figure out those connections, and so I guess what I'm saying is that there might be actually some benefits to it that are beyond what we're working on, that we're not considering at the moment.
And kind of on that note, this is leaving the realm of like science a little bit, not entirely, but if you were a PhD student today, what do you think you would be working on? It can be either in this field or some other field.
Let's see. It's a good question. I haven't really thought about it too much. I think for me, I've always, I don't really have a good answer for you for that. I'll have to think about it some more. Well, you can come back to that question.
Yeah, like if you, I guess to give like some hypotheses, it actually feels like when you were describing your original personality when you were back in high school, it actually feels like the type of mind that would have gone to physics much more than biology. Do you think like, and like it turned out you studied phylogenetics, which is kind of like a field with like lacks a lot of translation to the real world. Do you think it's like good for someone to focus on something that has like very theory heavy and relatively little immediate application? And the hope is that you can eventually convert that into like actually useful research, like during grad school or during like further research. Or it's good to like immediately start with the immediate like applicable stuff straight up.
It's a good question. So I think for me, I've always done things that I felt were more fun or more curiosity driven, I guess, research. I've never sort of sat down and said, okay, this direction is probably the most meaningful thing to do. It's more just like, okay, this is like a puzzle and there's no solution here. I'm just trying to figure out what's going on here. And I think when you go down the path of saying, okay, what's going to be the most, I don't know, going to make me the most money or something. I guess people are driven different ways. Like for me, that's not really what I think about when I think about thinking of a new topic to work on. For me, it's like, is there some unsolved problem here? And like, why does this work the way it works? And and I think I just like to solve puzzles is like maybe where.
So it's not even necessarily like I want to have this like grand scientific impact. It's just like I get nerd sniped by something and then I spend years working on that.
I think so. Yeah. Okay. Exactly. I mean, sometimes you're lucky and you happen to got curious about a topic that's actually turns out to be really popular and takes over. I mean, when I was moving into this whole evolution and coevolution field, I didn't think that this would be like some hot topic. So maybe I got pretty lucky in picking an area that happened to have exploded recently. I mean, there's definitely like a chance or like a lottery thing here. Yeah, like I mean, like for example, when I was moving into coevolution, a lot of people actually told me like, don't do this. This didn't work because people have been exploring this like since
Yeah.
the 90s and 70s and so on, and never quite worked. And so like say this is the wrong area to be in. But I think I still believe I could figure it out. And so, um, but it's also those area like people didn't think it's going to work at the time. And
Yeah.
so I mean, like right now, if I was to give somebody advice, I would say, well, it's I mean, if it's an area everybody thinks is going to work, there's probably going to be a bunch of people doing it because everybody else thinks probably the same way this is
Yeah.
going to work. And so it's almost more fun to say, okay, here's something that no one thinks is going to work.
Okay, that's good
It's
advice. Yeah, like not field specific, just pick something that like doesn't seem to have like billions of dollars worth of attention being focused on. So like, yeah,
well, I'm not sure if it's always the good advice. Like
that's right.
maybe ultimately you want to pick something that's not completely crazy, but um, yeah. But I would say who's ever listening to this, don't listen to me because I'm probably giving really wrong advice here. You probably don't. You probably want to pick on something that will secure your future somehow. But at least for me, I've never every time I pick a problem, I always think like it's more like let's explore it, like find something cool and see what we find. And sometimes you get some cool things that come out of these things.
Mm.
Yeah.
Do you think, I know you mentioned like you had this like passing interest in like pursuing history when you were in college. Do you think that like what would an alternative Sergey look like? Like if you had not like touched biology at all, what field do you think you would have focused on?
It's a good question. I mean, I do like I did find that I enjoyed history in terms of like understanding like what happened in the past. Maybe this is also why I was interested in philology because that's also history. Yeah, I'm just sort of making this connection now potentially. But one thing I found is that when I went to college and I started taking some of these history courses, I quickly realized I didn't have the right sort of, I guess you say, background because at least my colleagues were all like, they all like learned Latin at some point. I was like, I don't know if that. So I quickly realized, okay, this is not something I was pre-trained to be able to handle. And so, but I really enjoy just learning about the history of science because like what people have done before, how do people decide to work on DNA and so on and so on. That's kind of and so, but I think it's kind of fun to see it from that perspective. I don't think I'd be able to get into it now because I probably have to go back and learn Latin again. But
Well, like at least for like a history of science, you probably don't need to learn Latin. Do you think like you're like an active consumer of like the Genentech book that came out that explained like the history of recombinant insulin? Like, do books like that particularly appeal to you, and you enjoy reading about how like scientists in the 80s and 90s like really like worked on things?
Yeah, I definitely find it really exciting. Just thinking about like how to, because I think one thing that I found quite fascinating is as you sort of look at all these different stories of how people made breakthroughs, often it wasn't because somebody actually had a very specific thing they wanted to test. Like often it's like somebody does something completely random and they see some interesting unexplained signal and they start to pursue it and they get a really cool discovery. And so I think a lot of most important science have been sort of made that way. Like people sort of see a random signal and they pursue it. And that, of course, sort of brings up the question of like, how do you actually get this research funded, right? It's like if you don't have a direct question and you just want to like explore in the cloud and find something, but that being said, I mean, there is research for like, there's questions in terms of basic research. I mean, there's funding for basic research, what I'm trying to say. Like for us, often what I do is we try to propose like a general area to explore. And every time we start projects, we start exploring areas. But then if we see something interesting along the way, sometimes we change direction to that area.
When I think of your lab, I consider it much more from the outside an applied biology lab. In your head, do you still kind of consider yourself like a basic research scientist?
Yeah, I would say we're more. I like to think we're more basic in terms of like thinking about more theoretical problems, big picture problems. We do collaborate with a lot of people who do have specific applications. So, for example, some people want to do protein design, and we say, hey, maybe some of these tools or some of these recent hacks we found could be useful for protein design, and then we collaborate with people like that. but yeah, I like to think that we're more on the basic sciences. Yeah.
Is do you have a when it comes back to these like you pursue problems that you're often like just like feel innately like interested in and like it just makes you want to pursue it for years on end? Do you think you have a good sense of taste for what makes for a good problem and what doesn't make for a good problem? I remember reading this article a while back about like types of problems you should focus on during your PhD and like what makes for good, something good to focus on for the next 5 to 6 years. What's your own internal sense for what those types of problems are?
It's a good question. So I'm a disclaimer. I'm a new PI, so I haven't seen. I only have a sample size of one, me myself. So I don't know if these are good answers or not. It's. I mean, I guess one could say that. I mean, I think it's good to start with some problem, regardless if it's that interesting or not. Because I think you sometimes just need to get yourself familiar with the field, right? So like you start with something, say, hey, I don't know, protein docking, right? And so regardless if you're interested in that problem or not, but as you start to explore the different tools, you start to realize different limitations of these tools. And then something you might come across something interesting that you pursue further. And so at least the way I've currently been doing this with my current students, we'll see 5 years from now if it's actually a good idea or not, is to say, hey, let's just start working on something. Like here's some interesting areas that I find interesting, but with the intent that at some point we'll see something else. And sort of thinking back now, like when I look at all the things that we have published, I don't think there's been a single thing where this is what we thought we'll be publishing 5 years from now. It's always like we start with something and we're like, oh, we found something really cool along the way. And then we realize, hey, this could be applied to this and so on. And so I think it's just like getting started, starting to explore. And I think curiosity is probably a really important component. Like you have to, like you see something that doesn't make sense and you sort of keep pursuing it.
So you typically don't have like a 2 to 3 year plan for what paper do I expect to publish in 3 years?
Well, we do start with that. Like the idea is like you start off with a plan. It's like, okay, here's a problem. But I guess what I mean is like when you start that problem, you might end up deviating from that problem, and that's totally okay. At least the way I view it, it's like you start like you have a. I mean, I guess one in a maybe a more ideal world, one would say, okay, let's start with maybe it's good to have two problems, like one that's a little more safe. You know, okay, if you do these steps, it'll work. And then you say, and then you sort of have like maybe, I guess maybe similar to optimization. You have like I guess you could say, I was thinking more like, what is it called? Simulated annealing is like you have like different chains. Like you have sort of a fast moving chain, a slow chain, and you could potentially do some kind of recombination. But again, but this is kind of getting a little too, but I would say it's good to have a plan. So just in case somebody, but I would say don't deviate, but don't be afraid to deviate from the plan. Because sometimes like you might come across something cool or interesting or unknown, and it's okay to shift over and explore that unknown. And that's usually where the most interesting impact will come from.
Do you think it's generally, when you're setting up this initial plan to start off with, do you think it's good to focus on like incredibly ambitious, largely intractable problems, or it's good to start with like a pretty close-ended thing that, you know, if you apply like enough engineering effort, you will have a paper at the end of it?
Yeah, that's a good question. I don't think I have a good answer for you because I think right now I've at least any time I've done in research myself personally, I've never said, okay, let's if we do this, we'll get a paper.
Okay.
I was like, hey, let's explore this area. I know eventually we'll get a paper about something, but whether or not we'll get the paper for that specific thing we started with, and at least that's the way I've been doing science. Whether or not it's a good idea, if that's a good idea or not, we'll find out later. But I feel like these are kind of questions that might be more relevant to ask somebody who's been, I mean, PI for many decades. They could probably tell you, hey, I've seen all my students, and these are the things I experimented. These are things that worked and didn't work. But just coming from myself personally, I've always I always found like it's good to have a goal in mind, but deviating from that goal is going to be an important component to actually finding something really cool.
I imagine being a PI, you're relatively new PI. You've been around, I think, at MIT since January 2024.
Hmm.
And I imagine as PIs must do, you have to like specialize a little bit. Like you can't kind of be all over the place, especially early
Hmm.
on in your career. Do you think there are like areas outside of protein structure determination and protein binders that you wish you could have an extra 12 hours of the day to focus on?
Hmm. Hmm. Well, I would say me personally, I mean, we are somewhat protein, I guess, centric, but we are trying to expand a little outside of that in terms of thinking about genome scale related things,
Mm.
because ultimately proteins don't exist in isolation. But even for the same protein, the RNA itself might actually have other components there that determine the structure. So what I mean by that is, like, for example, like different codon usage could potentially influence how a protein folds, just because they might stall the ribosome in different parts. There might be some things up or downstream of the sequence that maybe changes influences the expression levels, or maybe even changes where things change. So I guess in some ways we are also exploring that other side
Mhm.
things. Yeah, yeah,
Do you think like your lab will start poking at like RNA and DNA models in the near future?
yeah, yeah. So we, for example, been exploring models like Evo coming out from the Arc Institute, trying to understand what are they learning? Are they learning something different? Um, but yeah, this is part of the, I guess you could say, um, I think we're kind of moving more and more and thinking about proteins in the context of a genome as opposed to proteins in isolation.
Okay,
Yeah, yeah.
what's your take on Evo 1, Evo 2? Like what are your general thoughts about it?
Yeah, it's quite interesting. So I mean, we've tried applying some of these techniques that we developed, like categorical Jacobians, to Evo,
Mm.
and we're currently not seeing that it's starting contacts in terms of protein interactions. We're still internally debating: is this because the approach that we developed, this categorical Jacobian, just doesn't work for these kind of models, being autoregressive and being like predicting single nucleotides, or is it because the model itself is learning some other signal that's dominating the protein signal?
Sorry,
Is
when
yeah.
you say that, are you saying like it's unlikely Evo 2 is actively relying on coevolutionary signal?
So what I mean is, like, so to step back a little bit. So when you do have a model that takes a sequence and returns a sequence, you can ask the question: as you perturb the inputs, how do the outputs change? And so if you think two positions are dependent on each other, when you perturb something, let's say in position 10, the logits or the outputs should change differently in position 10. So in position 1 versus position 10. And when you do this kind of perturbation experience in protein language models, we clearly are able to recover contacts. Like, see, these are interactions. But when you do the same thing with models like Evo and Evo 2, you see it's able to recover like RNA interactions, like RNA stems and so on. But we don't really see a strong signal for protein residue interactions. And so one hypothesis is like, well, maybe there's another dominating signal that's completely sort of washing out. Like, it's possible it's still learning contacts, but maybe like other forces at play. For example, like encoding start and stop codons, encoding the starts and ends of genes. Maybe that signal is much more powerful, and so it's kind of washing away the contacts.
Gotcha.
So maybe we just have to disentangle the signal. And so there, or maybe it's not learning any contacts at all. And so this is sort of still like an area that we're investigating. Um, I guess another hypothesis might be is say that maybe that the if you sort of look at any given codon, and you sort of, if you remove, for example, the third codon position, you could still recover what the amino acid is.
Mhm.
So, like, for example, masking the third nucleotide of a codon is not really informative in terms of learning coevolution because it recovers it. Same thing even for the first position. Like, if you mess with the first position of the codon, you often stay within either hydrophobic or hydrophilic. You're actually not changing the property of the amino acid.
So there's
Um,
something off about like the masking strategy that like Evo 2 is using that like leads to these like strange results.
Well, that's one hypothesis. Like, maybe the reason why, like, for RNA works perfectly because with RNA, you're actually every nucleotide, or I guess the ribonucleotide is interacting with something else. And so if you mask it, then of course you need to change the other guy to compensate it, or if you not mask, but if you mutate it, I guess.
Mhm.
But with protein sequences on the I guess you say DNA level, it's possible that maybe you could just recover a lot of the statistics of things that you've masked just based on local properties. Like, if you're in this codon, you know you're going to be an alanine, so the third one is not going to tell you much difference. Or if you mask or if you mutate the first one, you could look at the other two to figure out what the first one should be. Even the middle position, like for example, transition versus transversions, keep you in the same amino acid property.
Mhm.
And so maybe there's just a little too easy to reconstruct the masked tokens without having to actually fully understand the protein interactions.
Mhm.
Um, but that's something we're still investigating, so we'll have to see.
And kind of on this note of like potentially like off masking strategy for Evo 2, do you think like clearly like modern day biology models have a lot of pathologies and like edge cases where they don't quite work as well? Do you think the fault of that can be largely placed on like the internals of the model itself? Like the engineers and scientists need to like fix it? Or do you think it's often much more like a data problem?
Well,
Because I think for
yeah,
a long time, like the data problem was like clearly focused on by a lot of people. And it seems like you can actually get pretty big gains by just like messing around with the model, like which is what your lab does a fair bit.
hmm hmm Um, I mean, it could be that. So I guess another hypothesis of why we don't see context is that maybe the model just needs to get much larger.
Yeah.
So what I mean is like, if we do believe that all language models are doing is just storing information or storing evolutionary statistics, there's probably a lot of evolutionary statistics that you need to store about the genome, and suddenly there's just too much things to store, and so you're probably going to store some of the, I guess you could say, the low rank signals like conservation and so on, and then maybe as the models keep scaling, you might start to learn more of these sparse signals, which are contacts.
Mhm.
And so it kind of makes sense as like the first signal you learn is conservation. And then as you scale, and that's actually what we see, for example, with some of the earlier protein language models, like there was tape that a lot of times just learned like conservation. But then as we moved on to like ESM2 and you start to scale up, you start to see like, hey, now if you start comparing the different models that were trained, some of them don't learn any contacts and some of them start to learn contacts as you make them larger and larger.
Mhm.
So it's also possible we're just like at the very early stages, like Evo 2 is just like the very first early model, and that maybe it needs to be like 10 times bigger or more to finally start to learn those details.
Do you are you aligned with the idea of like DNA is all you need, or do you think at some point you need to bring in like structural information?
Um, it's, I mean, I guess one doesn't necessarily need to bring it explicitly because I guess the idea is that if structure is important, then the model, the attention would learn those structural constraints
Mhm.
implicitly. Like if that, like for example, if structure is important for reconstruction task, it should be able to pick up on that information,
Mhm.
and that's what we see with like ESM2 and so on. Um, that being said, there's possibility that you could unify some distance space by introducing structure.
Mhm.
And so what I mean by that is like, for example, people have trained like SA Prot from a group in Westlake, and then Pros T5, for example, they started introducing structural information into language models. And SA Prot team, for example, found that sometimes you could do much better at predicting effects of mutations by introducing structure. But the structure resolution is so low, like they're using 3DI tokens from Martin Steineger's group. And so it's kind of like, if it's the resolution is so low, how is it able to like use that information? And so one of the current hypotheses, like, well, maybe what's happening there is that you currently, like maybe let's say in the language model space, you learn that this is protein A, this is protein B, and they're far away from each other.
Mhm.
But in reality, they might have really similar structures. And by maybe introducing even some really low resolution structural information, you sort of start to align those spaces together.
Mhm.
And so maybe the model sort of learning different categories of proteins, and those spaces are not well aligned to each other, and you're sort of bringing them close together. And so now you can borrow statistics from neighboring families that you didn't borrow from before. And so I think structure could help maybe sort of learn sort of maybe a better latent space of protein sequences.
Mhm. So it's like
Yeah, yeah.
it feels unlikely that like a single modality will like dominate. Like there'll be like a single like multi-billion parameter model using only one modality of data that'll dominate every single benchmark.
I mean, theoretically, it should be possible. Like, it should, like, technically, DNA should be all you need. But in practice, I think if you don't have a large enough model, or maybe if you don't have enough, or something wrong with like maybe you get stuck in a local minima. I think sometimes structure could help steer the training in the right direction.
Mhm.
But theoretically, it shouldn't need, like, you shouldn't need it, I think.
Yeah,
Uh,
makes sense.
yeah, yeah.
And one question one of your old students actually told me to ask you
Okay.
is first context for the question. Making test train splits for models in protein engineering is really hard since there's so many possible ways to leak data. You need to simultaneously think about homologous sequences, structure similarities, and functional similarities between proteins in your test set and your train set. What do you think most papers get wrong about splits in this field, and how could it be improved?
hmm Well, I think one of the issues is that uh, we have it's not an issue. Like I think it's good. We have lots of people coming from the computer science world into biological problems. And so I think it's great that a lot of people are coming and helping us solve all these problems. And so you do have to sort of consider the relationship of things. But even when you do consider the relationship of things, like where do you set the cutoff? And so this is where the debate in the field is like, well, you can use sequence identity of 30. And this is usually people say, but where that number 30 comes from is usually people say if it's 30 or higher, it's probably doing the same thing. But you can still have things less than 30 and do the same thing. It's more like 30 is more of a cutoff of something being positive, but it's not a cutoff of something being negative. And so I think people often like saw the number 30 and they're like, okay, we're going to split at 30% identity. But in fact, you could have proteins that do the exact same thing, have the exact same structure, and with sequence identities as low as 10%. And so that's where then you say, hey, I split by 30, but in reality, you still have an overlap in the trained test set. And I guess one could say, well, let's use structure. Like, let's try to use structure here. But the problem with structure is that you also have a similar problem where maybe in one organism, there's domains that are in different orders. So for example, you might have the exact same, essentially, I guess you could say protein, but this protein has different arrangement of the domains, or has a couple more extra domains, or has some disordered loops or something. And then you compare those two things and say, hey, they look different, but in reality, they have some distantly related relationships. Because any given protein is actually made of a bunch of domains, and these domains could be in a completely different order between different organisms. And so if you do a structural superposition, you might also be misled to think that they're different.
Much more different than they actually
are. Yeah. So it's almost like you first have to split by domain and then compare domains. But then the problem is that within domains, you could also have rearrangement of secondary structures. Yeah. Like you could have the same exact protein, but maybe the order of the secondary structures are actually in different orders. And so that creates a bit of a problem. Like how do you but then at some point, if you start to, like you say, well, let's just split by amino acid, and then it's like, well, everything's related. So that's kind of I think one of the tricky things is you have to be a little careful in that space. Like you have to, it depends what your claim is though. I think, for example, to step back a little bit, I mean, before deep learning, there's always been a field of remote homology search. Like say, hey, if I could find a similarity between one thing and something super distantly related, like less than 10% identical, and I could say they're the same, it's actually still a very, very important problem to work on, because you could say, well, if I can find remote similarities, maybe I can make hypotheses about those things. Say, if this looks like this, maybe this also does the same thing. And so there is benefit to remote homology search. And so if you say, hey, I have a model that does better remote homology search, that's actually okay. But if you say, hey, I have a model that works because it learned physics, then suddenly now you have to be more careful how you're going to split your data. You have to say, I have to completely make a huge giant hole in the sequence space and say, this is still generalized there or not.
Do you think there's, I haven't actually seen that many people do this, but I've thought in the past, like, why can't you just use like the embeddings of like a language model as like a way to stratify things? Is there like some pathology there, like as to like why that actually, like as like the issue you point out with domains, is there some fundamental issue with like relying on embeddings?
Exactly. So it turns out it's because of the domains.
Okay.
Like,
okay.
So if you take the embedding and you average it, you've essentially already lost the information about what the order of the domains are.
Okay.
In some ways, you could say that's a good thing. Like maybe you don't want the orders. Like if you average, you got rid of the order. But the thing is, sometimes they would have in a scenario where in one organism, the domains might actually be in a different protein. Like there could be 3 proteins coming together with different arrangement of domains. In another organism, it could be one protein with all those domains stitched together. Like all the domains are fused in one protein. And so if you average, all those things will look different from each other. It's almost like you want to first take the original embedding and chop it down and then compare all the different chopped segments to each other. But as soon as you start to do that, then it becomes then you essentially are back to aligning sequences. And if you start to aligning, then if they're in different orders, you can't really align them anymore because you can't use dynamic programming. And so it creates a bit of a challenge there.
Yeah, like I like the way you've set up that dilemma, it feels unsolvable. Like, like what do you think researchers should be doing?
Well, like in our example, like for example, we had a recent paper with Nick Polizzi where we had this paper AF to bind, and so there what we did was say, you know what, let's make sure there's no overlap on the PFAM level, like the protein family level based on sequence HMM comparisons. Let's make sure there's no structural overlap, but also let's cut out the actual binding site and make sure there's no overlap in the binding site. And so we went through levels and levels of trying to sort of say make sure there's absolutely no similarity between these two things. And so then we were more safe to say, okay, these are probably not. But once you start doing that, like for example, for protein small molecule interactions, I think we found total there's like only 500 proteins at the end of the day. Like I mean, like independent samples that don't look like each other anymore.
In terms of the binding site?
Yeah. Well, so it's like if you start to go through, say, let's cluster based on sequence, structure, binding site. Turns out there's not that many examples anymore.
Gotcha.
But of course, you don't have to remove the data. Like you can still keep the members of that cluster and within your training, but it quickly realized there's not as much data as you used to think there is.
Do you think the stratification problem also pops up with DNA language models? I didn't look too closely as to like how the Evo team stratify things. I'm kind of unclear how DNA language models work in this capacity.
Yeah, I think for language models, people are not as concerned in terms of train test split. But what I mean by that is like there is concerns like you definitely don't want to just to memorize all the sequences. So you want to definitely remove some sequences to make sure you didn't memorize. But I guess if we come back and think about if language models just storing protein families and their evolutionary statistics, then in some ways you don't actually want to remove sequences from an entire protein family. You want to remove just enough to confirm that you're not just memorizing sequences, but you don't want to remove enough that you sort of obliterate an entire protein family from the training.
Well, the hope is that there's like transfer to like an unknown protein family. I guess this is the whole like ambiguous versus non-ambiguous thing.
Yes, exactly. So I think you're right. So if you want to make the argument that the protein language model has learned sort of a new space, then yes, there you want to actually make sure you remove anything that's anything remotely similar to it. Yeah, but if you say, hey, I just want a language model that sort of stored all the information and statistics of all the protein families, then in some ways you don't actually want to remove too much information. Like you kind of want it to see sequences from every single protein family because you want to learn an embedding of all these protein families. So I guess it depends on like what you're using the model, what claim you want to make for those models.
And I imagine if I'm like to infer your viewpoint correctly, is it that like do you think like trying to train a model such that it'll be able to like divine like a new protein space entirely? Is it like new protein family entirely? Isn't it actually all that fundamentally useful? Because like most of the known protein families are like in the space of things we actually care about.
I mean, I would say it would be very useful.
Okay.
I mean, ultimately the goal is to be able to have a model that can generalize to all proteins. And so I think one example of plots that we like to make and other folks too is like the number of sequences versus the performance of the model. By number of sequences, meaning how many sequences are there in the protein family. And currently, like most protein language models, I think all of them actually have like a curve this way, right? So it's like if that family doesn't have that many sequences, the model has really, really poor performance. I mean, eventually we'd want the model just to, I mean, this line to be flat. It shouldn't matter how many sequences there are in that family. And one could imagine keeping, like, I guess you could say, hiding some sequences, or one could just alternatively just keep evaluating if that curve is shifting.
Yeah.
Like if that curve becomes suddenly flat, then you're like, okay, it's learned something interesting, fundamental. But this also sort of brings up the question, sort of a fundamental difference in how sometimes people in the computer science world think about problems versus biologists. I think sometimes when I talk to my computer science colleagues, and I tell them, hey, we want to predict things out of distribution. And I think they get a little bit like what it's like that's sort of goes against the fundamentals of machine learning. Like you want to learn things within distribution, and you want to be able to sample things within distribution. And the thing is, in biology, often we want the out of distribution things. And so, like, how do you get a model to go out of distribution? And I think the things that I find recently more exciting is like, well, things where you could iteratively reason over things might be a way to sort of move into that space. But
Like going back to this
Yeah,
idea
yeah.
of like many different seeds and like seeing where on the fitness landscape the model places you.
Yep, exactly. And I think there's been in other fields, for example, people are now introducing like chain of thought and so on, where maybe now if you have a model that says, hey, return something in one go or one, I guess say one shot or a zero shot, it's sort of, it goes through and makes the best guess it could. But if you sort of iterate on that, maybe you have it be able to explore outside of its training. And in some ways that you could think of that, that's what essentially Alphafold's doing. Like Alphafold say, I'm going to make a guess that's like zero recycles, and then you iterate and you sort of move around. But maybe if you do many independent seeds, and I think that's what some of these models like O1 and O3 are doing. Like they have many independent starting points and they explore. And so I think in some ways, I guess we could say we've been already doing that for a while in the protein world, and they're kind of catching up. But I guess one could also say, well, maybe some of the techniques learned in that space could be applied now in the protein world as well.
Do you think there is that much utility in like integrating, like instead of having like actually more straightforwardly, do you think there's useful information in integrating like wet lab assay data when like I guess like one example of this is like introducing binding affinity into like Alphafold 3. And
Mhm, mhm.
instead of just having like two protein structures together, you also say like, oh, this is the KD that came alongside it. Do you think there's that much value in doing something like that? Or empirically, it doesn't turn out to actually be all that important?
I think in practice it could be useful because like right now the model, like for some methods like AlphaFold, assume anything you give it is a good thing.
Yeah.
Like it's a real thing, it's a real protein. But turns out in some organisms maybe you want it to have higher affinity, in some organisms you want less affinity. Like if you're in a hotter environment or colder environment, maybe you want the protein to be less or more stable. And so like right now, the model just assumes everything you give it is a good thing. And so it's sort of learned some interesting average of all that stuff, but be able to sort of tune that label during prediction or something, or during design could be actually useful.
Do you think there's anyone trying to integrate like almost like human labels in like because like alongside a lot of these protein structures that are like deposited in the PDB, there are some like semantic labels that go alongside it of like this is a particularly like well resolved structure or like this residue was like
Hmm, hmm.
hard to resolve. Do you think integrating that sort of information, like bringing in semantic level information, is actually all that useful, or is it just kind of like so far beyond like what anybody else in the field is focusing right now that's not worth poking at too much?
I mean, I think it's a definitely exciting direction to look into. I don't know if anyone's tried that yet. I mean, people have tried filtering on that
Mhm.
labels. Like for example, I guess the most recent example I can think of that was really cool is the soluble NPNN
Yeah.
paper, where folks said, "Hey, let's retrain protein in PNN, but only on soluble proteins."
Mhm.
I guess a newer versions of that is say, well, what if you just provide a label so is this protein soluble or non-soluble?
Yeah.
Like right now, the model sort of implicit, like the default model, like protein to PN and even AlphaFold,
Mhm.
by default sort of sees that sequence and maybe tries to infer as it's soluble, non-soluble. But now if you retrain on those that kind of label and or introduce this label, you could essentially now tune and say, hey, this is a protein that's in the membrane, and maybe you should be folding it differently or filtering the constraints differently. Um, so I think there's already been demonstration of this in the context of soluble in PNN.
Makes sense.
This is Hustis and Bruno Correas. And so, yeah.
Is kind of on the subject of soluble MPNN. One question I realized I should have asked much earlier is what are your thoughts about sequence structure co-design versus like one step structure, second step sequence?
Um, we recently, so Yiling and my group, we recently put up a preprint where we sort of explore this a little bit, where we co-design both at the same time.
Mhm.
And our current hypothesis is that there is actually a benefit to co-design from the perspective that, uh, like for example, let's say if you start with a structure and you come up with a sequence for it, maybe there's a better structure that can encode even a better sequence. And so having ability to move the structure closer to the sequence space and then finding a better sequence, there's some benefit to that. Because maybe there's like it's possible for this fold that you could find even a better free energy sequence. But if you unless you see that structure, you won't be able to come up with a sequence.
Mhm.
So I think there's benefit in that space. But it doesn't necessarily solve the problem of um, uh, does the sequence fold into that structure, no other structure. So you still need to to have the, I guess you say, the structure prediction module in place. So I guess there's been some work by, I believe, like folks like Jason Eam at MIT, where he looked at, say, hey, could we co-design things? But I feel like when you want to co-design things, what you really want to do is you want to co-design sequence structure and folding.
Yeah.
Because the way I like to think about it is like when you're designing a protein, you're not designing a sequence or structure, you're designing essentially a folding landscape.
Yeah.
And one of the problems, so it's not about co-design of sequence and structure, it's co-design, I guess you maybe try design, I don't know if you want to say it that way, of sequence, structure, and folding. And it's kind of hard to think about how the models like diffusion could incorporate folding, because they're considering that one structure, but they're not considering the ways that protein would fold into that structure, or how a protein sequence would fold into that structure during the process. And it's not super clear to me how you would actually incorporate that at the moment.
Like as opposed to pure structure models.
Well, I guess there have been approaches where people say, well, let's diffuse the sequence and structure at the same time. Yeah. And then the question is like, are you both satisfying the folding and inverse folding? And so there I would argue like, no, you're not satisfying inverse folding, because inverse folding requires that you come up with a sequence that only folds into one structure and no other structure. Mm-hmm. And so unless in your diffusion, you're somehow accounting for all the other ways the sequence can fold, you're not actually optimizing for the inverse folding problem.
I guess like protein MPNN is trying to satisfy that.
Well, this is what I would argue. It's not really yet. Okay. So I think there's been unfortunately in the field, people have been using the word inverse folding incorrectly, because formally inverse folding, previously, and I think folks like Kendall have defined it as like finding a sequence that folds into one structure and no other structure, but also at the same time making sure it's actually accessible. Mm-hmm. Like, because you could imagine finding a sequence that
like thermodynamically accessible. Or
I guess I was thinking more on the kinetic side, but what I guess I mean is like during folding, you want to make sure it fold, like for example, there's not a huge barrier. Yeah. Unless you have a chaperone, maybe that's one way to lower the barrier. And so that's what formally inverse folding was defined as. I think, unfortunately, folks in the computer science world, when they're like, hey, let's develop a method that takes a structure and returns a sequence, we're just going to call that inverse folding. Mm-hmm. And I mean, it's a nice term, but unfortunately, it's not actually the real inverse folding from what as the term was originally defined. Mm-hmm. And so I think in the protein and PNN, people were pretty careful not to call it inverse folding, but I think other folks who've developed similar like methods have called it inverse folding. Mm-hmm. And so I think at least on Twitter or X, I always, every time somebody says inverse folding, I try to correct them and say, hey, this is not really inverse folding. You're not actually satisfying the inverse folding question, I guess you could say.
I guess, but like if you use that definition, no structure to sequence or sequence structure co-design method is satisfying that metric.
Well, this is what I would argue. Inverting a structure prediction model is sort of maybe implicitly trying to satisfy that condition. Mm. So like you say, I have a method that takes a sequence and predicts a structure, and if I invert that model, then as you're changing the sequence, you're essentially every step along the way implicitly checking, are you satisfying that condition?
Gotcha.
And so I'd say that is closer to inverse folding than a method that takes a structure and tries to predict a sequence from that.
Gotcha. And so one thing I am still a little bit unclear about is, why do you think that structure sequence co-design models are not able to do this?
Well, because they're not at any point evaluating along the way if that sequence won't fold into something else.
Gotcha. Whereas the sequence to structure model is during its training process, like doing that. And then if you invert it, like you hope the one-to-one relationship stays the same.
Kind of, exactly.
Gotcha. Okay.
Now, I mean, people who actually study protein folding might tell you, okay, no, that's not true because the model doesn't explicitly fold proteins. Yeah. And so maybe you do actually need a model that does protein folding first to be able to do that. But we think it is sort of an implicitly maybe pushing in that direction.
One of the last questions I had is, you've discussed a lot on Twitter about recent funding cuts
Mhm.
to like kind of like academia in general, and specifically like concerns about your own lab. Um, One, like I kind of want to like talk about like the best case scenario of like, let's say you did have a few $100 million you could spend on any type of basic or applied research you wanted. What would you want to work on?
It's a good question. I think for me, if I had like lots of money, I would try to think of if there's some kind of experiments we could do, like wet lab experiments, to collect information about protein folding.
Hmm.
I think that's one of the things. Like right now, we have absolutely no experiment. I mean, there's been people like trying to remember his name. It's a guy in Canada. It's not Louis C.K. It's like K. Lewis or something.
Mhm.
Um, okay, we will have to. Okay, maybe I'll start with that question again. So I think um, I'm trying to think of a good way to explain this.
When you say
I-
like measure protein folding, is that like NMR level, like the actual process of the protein
yeah, yeah.
folding? Okay,
Mhm,
okay,
mhm.
okay. And you would want to just like collect like thousands of these results.
Yes, yes.
Okay.
So I think one of the problems right now is like we have. So let's say we okay. So let's just say we step back and say we want to solve the actual protein folding problem. Um, and by protein folding problem, what I mean is like starting from an extended chain, find the right conformation of the structure, step by step,
Mhm.
and actually thinking about all the steps that it required to get there. One of the issues that we don't actually have any truth, like we can't really train a such a model. Um, I mean, there were experiments where people look at transition states. That's the researcher I was trying to remember the name of, Louis K. I think his name is. And that but that just gives you for just like one or two proteins. I think they did some NMR transitions that they've measured there
Mhm.
that are sort of what they believe to be intermediate folding states. And so I think if there was lots of money, I'm wondering if there's ways that we could develop experiments that could actually measure a large scale, like a bunch how different proteins fold. Um, and I've been sort of thinking about how we would go about doing this, and it's a little tricky because it's like you have to kind of measure it on individual molecule level to some extent,
Yeah.
because it turns out every protein will fold differently. So I don't know um, if there's a one particular way. Maybe there's some ways to synchronize it. Like for example, people have done these like molecular tweezer experiments,
Mhm.
and so in some ways you can unfold and refold the protein by pulling on the NNC terminus. If there's some way we could image those things.
Like just like a line of amino acids, the final state.
Yep.
Gotcha.
I mean, I mean, the other problem is also pretty exciting. Like, hey, how do you extract all the different dynamics a protein could be, or maybe all the different conformations it can fold into? But I guess what I'm saying is like, it'd be great if we could somehow actually get step by steps snapshots of the structure of folding.
Mhm.
And so if I had money, I think I'd throw it all on that.
In some
And so
sense,
if we
is
could,
that not dynamics?
it is dynamics, yeah. But usually it just happens once and it stays there.
Yeah.
And so then with NMR, you're kind of stuck and you're just watching that one structure vibrating and moving around.
Mhm.
But the question is like, could we sort of take snapshots of it going from an extended down to the final structure?
Do you
Um,
think the like let's say we did have like a perfect NNP that was capable of taking this like string of amino acids and seeing like femtosecond to femtosecond, how does it like fold into a like a final structure? Would
yep.
you still want this NMR data set? Is there something useful about measuring it in like the real physical world that you cannot get from a simulation?
Um, I mean, if we do have a good simulator that could actually do the whole process,
Mm.
I mean, I guess I was thinking more like, could we generate data to be able to train a better simulator to be able to do this?
Gotcha.
Because
Okay.
I guess I'm under the impression that the current, even if you had infinite compute time and an MD trajectory, might never have found the solution,
Mhm.
just because there's sort of inaccuracies in the energy function and so on. Um, but if we assume that everything's correct in MD, and then you're right. I mean, if we have an infinite compute, we just do that.
Yeah. But like, yeah, I think
Yeah.
empirically you are correct in that they're nowhere near that place of being able to go from.
Yeah. I mean,
each
I think D Shaw showed like they could do like really small domains and they can get them to fold up. But that's we're quite far from that. I mean, I think we could approximate a lot of this with neural networks, but we sort of need some of these intermediate steps that we could train on.
Mhm.
And so I think that's. But the question is like maybe like why would you even need this? Like if we already have methods that can predict a lot of protein structures,
They're like end states. Yeah.
like the end state, right? But I guess I'm under the impression in order to be able to predict some of these really complicated protein structures, it would be good to be able to see some of those intermediates, because maybe you actually want to push the model to explore those steps step by step.
Returning back to the NMR research approach, are you currently pursuing any like attempts to like really scale up wet lab efforts in your own lab, or are you largely right now entirely focused on computational work?
I guess just to clarify one point, I wasn't saying NMR was necessarily the solution to the problem. I was just saying this is one area that may be able to get there. Um, but besides that, so we're not trying to do NMR right now. Hannah Wayman Steele is actually starting a lab where they're going all in on the NMR stuff. So she'd be a cool person to talk to at some point. But at least in my group, we've always thought of it more. I've always thought of it as more of a sort of theoretical computational group. But that being said, we have started some efforts in the space of sort of building up maybe some robots that can do experiments for us, like maybe some automated lab kind of things. But this is just more of the exploratory phase at the moment. But primarily we're a computational group and we collaborate with people.
Do you think there's that much value? Like you're really interested in the like start like the pathway from like start to end protein structure. Do you think there's that much further value in like mass collecting like end structure data like further? Like let's say like we double the PDP overnight. Do you think it's actually would make these models all that much better?
I think they could. So for example, there we have, we have a lot of structures for prokaryotic organisms, and we can now predict a lot of prokaryotic organism proteins. And because we have like lots of sequences, we can get coevolution from them. But I think one of the challenges has been for eukaryotic organisms where you have multiple domains, and we still don't quite know how those domains come together. And often when you make a multiple sequence alignment, they would cover like one domain, but not the other domain, or separately cover each domain. And so then when you give these to Alphafold, it sort of arbitrarily places these domains in 3D space. And so these sort of large multi-domain proteins is areas where we actually have no information how they come together.
Gotcha.
And so I think if we, but besides that, there's also a lot of proteins that are not multi-domain, but only have a few sequences. And there's sort of a debate, like, are these mostly disordered? That's why we don't have that many sequences. Or maybe they actually are structured, or maybe they're only structured upon binding to another protein. So I think there's still a lot of proteins that it would be useful to have, especially for this problem of trying to go from single sequence to structure, where we have no evolutionary information. And so the more we can collect of those, the more we can start to create data sets to be able to maybe start venturing into this problem.
So if you have the choice between just like randomly doubling the PDP overnight versus like doubling it in the areas where like proteins are protein structures are either very large or they're suspected to be disordered, you'd prefer it in that latter category?
I guess I said both, but I think for me personally, I would say, for me, the more exciting part is like, well, regions that currently have really low PLDT in Alphafold, that might actually be ordered.
Yeah.
Like we think they're disordered maybe because there's a lot of correlation between PLDT and disorder, but I think a lot of that low PLDT is not because of disorder, but because just this lack of MSA information. And so I think if someone had like infinite money to give us, it would be kind of great to say, let's go ahead and try to solve all those structures that we think are actually ordered, but have really low PLDT.
Gotcha.
And in some ways, people used to do that back in the day with like structural consortiums to say, hey, if there's no homologs, but I think now we can use Alphafold to quickly tell us, okay, what problems are worth maybe putting efforts into solving.
It's like triage and like decide like these are the structures I actually want, and everything else is like information that the model already kind of implicitly knows.
Yeah, I guess in some ways, it's almost like active learning, I guess you'd say. Predict everything UniProt, look for everything that Alphafold failed on. Those are the proteins we should be trying to predict the structures for. But that being said, I think we just have to be careful because there's cases where the PLDT might appear to be high, but if you start to look at the PAE matrices, the predict alignment errors, there might be not information how the domains come together. And so even cases where we think we know all the domains, it would be great to know how they come together. And I think those are also useful problems. This is
one thing I've been again, a little bit of a deviation from what we've been talking about. One thing I've long been confused about is that there's this like implicit trust in what Alphafold 2 is confident about and what it's not confident about, or like what it claims to be. And it feels like people don't actually seem to run into adversarial optimization all that often. Why is that? It feels like in almost every other ML field, if you try to like really trust the model in like one specific metric and just keep like doing that over and over again, you eventually run into like adversarial edge cases. But that doesn't seem to happen with AlphaFold too. Is there like a strong like? Is there a reason you suspect why?
Um, I think it depends on the problem. So for example, there have been people who have been showing that, for example, if you try to predict protein peptide interactions, you get a lot of false signal in terms of you can have really, really high PAE at the interface, but the prediction is actually completely wrong. Like you have really high, um, but I think it depends how you got there. And what I mean by how you got there is in some cases, like for like one thing we saw recently in some of our work is if you try to score things with AlphaFold and you use a bunch of recycles, it sometimes gets confident about things that are completely wrong. But if you dial down the recycle, like go to recycle 0, you actually see that it's actually a bit more correlated with known measurements, because the question is like, did AlphaFold think they interact because there's some evolutionary information or maybe some motif matching, or just sort of reinforced itself and now it's really, really confident.
Yeah.
And so, I mean, it's true that a lot of times the confidence are pretty good. But I think the reason why people do trust it is because often they're not subjecting AlphaFold to sort of unknown problems. So what I mean by that is, often when people say AlphaFold predicts something, they already know these two things interact, or they know this protein folds into a structure and they want to know, what does it actually look like? How do they interact? Where it fails is when you don't know if they interact and you're asking the question like, hey, do these two things interact? And sometimes they will give you the false impression that they do interact. And I think part of the reason why this happens is because AlphaFold was only trained on positive data.
I was about to mention this. Like,
Yeah.
like what does negative data look like in this case?
Well, so negative data would be like, let's say if you purposely take, hey, two proteins that should not interact, can you fine tune methods like AlphaFold or Zetafold? There's a researcher, Chen Kong at UT Southwestern, that has been actually exploring this a bit. I think they had a preprint recently where they say, hey, let's purposely mispair things and then tell AlphaFold that these should not interact.
Yeah.
And she saw her group saw some success in that area where they could potentially try to now do a better job at picking up what does and doesn't interact. And I think that something like that could also work in the design space, where right now, any sequence you give AlphaFold, the assumption is that this thing will fold into a protein.
Yeah.
And so even if the sequence is like very suboptimal, it will find a way to sort of internally fix itself, which is great for structure prediction. Like you want a model to sort of maybe only see a few key residues and make the right prediction. But for a design problem, you actually want it to be sensitive to point mutations. Like you want to say, hey, if you put a hydrophilic in the core, it should not be predicted well. But it kind of assumes everything you give it is actually a valid answer.
Yeah. That's a cool idea. Has anyone like shown that like you said like this one UT Southwestern professor, it feels like it hasn't really percolated to the field at large. Do you think like long term there will be like positive sets and negative sets for like structured data, or it's like unlikely that will be the case?
And I mean, I would assume that people are going to be doing this more and more. I mean, I think for structure prediction doesn't seem necessary because like everything is positive.
That's
right.
Fair,
But I think, for example, for people that do care about PPI prediction, like what's interacting with what, I mean, this is why part of the reason Chen Kong was doing this, because she actually wanted to know, hey, which protein interacts with which protein, in which case she saw a lot of false positives from just looking at the confidence metrics. And so she tried to fix that problem there. But I would imagine in other cases where maybe you want to say which ordered or disordered region actually folds into a protein, there might, like, for example, people do see, for example, with AlphaFold 3, you give it regions that are completely disordered and will still predict a bunch of helices. And that's where some negative training could be useful. And in some extent, they did that. Like the DeepMind team did do that. They actually took AlphaFold 2 predictions and used that for training to say, repeat the same extended loops and so on. But it turns out there's some kind of mode thing where if you make the protein too large, it sort of snaps back into making helices everywhere. And so it didn't completely work out the way they were hoping.
Has I forget. There was a really good reason why they needed to use AlphaFold 2 prediction to like bootstrap AlphaFold 3.
Well, the issue was that so the problem with diffusion is that, or at least training with diffusion, is that it sort of learns some distribution about how things tend to interact with each other, and it's only trained on sort of order things, and so its prior is to make everything ordered. And so one of the things was like if you start to use
like it's like unable to be not confident about anything.
it's still not confident. Like if you look at the confidence metric, it's still not confident. But I think the problem was that a lot of researchers sometimes don't even look at the confidence. They look at the structure, say, hey, if it's giant, long, disordered loop, then it's probably not confident. But the problem with AlphaFold 3 was, at least before they did this sort of trying to correct it with AlphaFold 2 predictions, is that every single prediction always had helices.
Yeah.
Like anything you give it, like completely random sequence, it will always put secondary structure. And so then they're like, okay, how do we fix that? Well, maybe we could try to get it to reconstruct how AlphaFold 2 used to do. Like if it was not confident, it was sort of extended out into a loop.
Yeah.
And so they started training on AlphaFold 2 predictions to try to get around that problem. It seems
like
a little hacky, but
it feels like a bizarre design. Like there surely has to be a better way to do it than like just bootstrapping from AlphaFold.
I'm not sure how you would do it. So I think the problem is diffusion. The problem is like you need it to give you the wrong answer to tell it it's doing something wrong.
Yeah.
But the problem is it's always like the last step is always the right answer. And so I think with like structure module, like you can have it have, I don't know, the loop get clashing with everything and you add like a clash loss.
Yeah.
But like how do you add a clash loss in diffusion?
That's a fair point. Yeah, I guess like having this like physics-based oracle would like help things a lot.
Yeah. Well, it's like, it's almost like you want it to move into the, so I think this is why they had to put in the AlphaFold 2 structures in, because it's like now you train it, say, hey, if there's no information, extend it. And it worked to some extent, but it turns out if you make the protein too large, it just starts making helices again everywhere. So it's sort of learned to do that for small proteins, but not for very, very large proteins.
That makes sense.
Yeah.
And I think I am largely out of questions. Was there anything else you wanted to talk about?
I think we're probably good.
Okay, cool. Thank you. Thank you for coming onto the show, Sergey.
Of course. No problem. Glad I can chat.